Hello, I'm Masaki Miya from Japan, working at the Natural History Museum and the Institute at Chiba. And today, I'm going to talk about my fish eDNA metabarcoding. New biodiversity monitoring method enables simultaneous detection of multiple fish species from observable seawater. First of all, you may be wondering that what is eDNA metabarcoding? And you may say which species lived. This is very simple but a difficult question to answer. Visual observation or capture-based sampling need long time and can be very expensive and require taxonomic expertise. And a recent study demonstrated that fish leave a trail of DNA, which is called environmental DNA. Environmental DNA is shed from microorganisms, the feces or mucus or some substances, not DNA directly itself. Okay. This is eDNA schematic presentation of eDNA metabarcoding, which is beginning from water sampling, which is captured through a syringe and a filter. We can collect eDNA on the filter, from which we can extract eDNA, which can be subjected to normal molecular analysis after that. The most important thing is library preparation, which means we amplify target DNA fragments to which we can analyze by the high throughput sequencing system, which is called next generation sequence system. We can analyze the data by computer, and finally we can obtain the taxonomic table. And this is aligned DNA sequencing from 808 AD species, and the row is DNA sequences from fish, and the column is DNA, which is has a different color, four different colors. You can see that there is a hyper variable region which can be used to distinguish species, which you can see there is a conservative region at both sides, and we can design universal PCR primer. And I have designed MyFish universal PCR primer, which is called MyFish. And I have published MyFish primer in 2015, which is has been downloaded more than 400 times since the publication, and cited by 287 times. And this is a schematic representation of the library preparation. We employed a two-step PCR to amplify and append various adapters. In the first round-tail PCR, which amplified the region of interest, the primer consists of the primary region and the overhang adapter sequence used in the second PCR and pair-length sequencing on MySeq. The PCR cycle is 35 to 40 cycles. We can obtain on an average of 170 base pair. And second round to PCR to add index and sequencing adapter. You can see that the red portion is index, which consists of 8 nucleotides and different combinations. So we can simultaneously analyze as many as hundreds or thousands samples simultaneously. The resulting PCR product consists of the region of interest, which can be used as a species detection, and the MyFish universal primer, and the sequencing primer and index in both sides and closer fixed region. And this map shows the geographic distribution of case studies using MyFish primer. You can see that the MyFish primer have been used in 6 continents from Arctic, South America, North America, Africa, and Eurasia, and Australian continents, as well as 3 oceans from marine, the freshwater, the brackish water ecosystems. MyFish performance of MyFish primer has been evaluated by UK researcher, and MyFish. You may be wondering that the CO1 genes have been used for barcoding, but CO1 is not useful for eDNA metabarcoding. And this graph, 5 graphs, shows that the number of species detection from these waters in United Kingdom. The vertical axis is the number of species detected, and the horizontal bar is primers. You can see that a MyFish primer can detect 2.2 to 2.6 times higher detection rate than the CO1 primer. So we can use MyFish primer for deep sea organisms as well. And we demonstrated that we validated the performance of MyFish primer in aquarium tanks in Okinawa Chirami Aquarium. There is 4 tanks which harbor pelagic organisms, tropical fishes, and deep sea fishes, and mangrove fishes. I'm going to talk about only CO tanks. You can see that there are 60 species kept in the tank, which has reference sequences. And we can detect 63 species, which means more than about 90-70% fish can be detected by MyFish eDNA metabarcoding. Based on this proof of concept, we can analyze deep water eDNA from deep water. The problem is that deep sea fishing is very sparse compared with coastal fishes. So I have developed filtration systems that allow more than 50 liters of the water. And we use Sterilix cartridge for that purpose. There are about 15 deep water pumping facilities in Japan, which are aimed at generating power by using differences in the temperature in the surface water and the deep water. One of which is Kumejma Island Deep Water Research Institute. This is Kumejima Island. This is Okinawa main island, southern Japan, which is located about 27 degrees north. You can see that there is a very steep slope, and of the about 1 kilometer, there is a depth of 612 meters, from which we constantly pump up the deep water. And we have performed 4 surveys from December 2017 to December 28. For each survey, we used a total of 9 cervix filters, each of which filter 50 liter, and 3 of which are filtered simultaneously. We have performed EDNA metabolic coding, and we detected over 150 deep sea fish species. The most constantly occurred species is this one. It's similar to orange raffi, which is followed by semi-formed fishes, and you can see other fishes. And most distinct characteristics of EDNA metabolic coding is that there is no size selection. We can detect these big fishes over 3 meters, and these are small midwater fishes, which is only a few centimeters. And the number of species is almost constant throughout the survey. And it is very interesting to see that there is differences in species composition plotted on the two-dimensional figure. You can see that, for example, there are 2 December samples from 2017 and 2018, but these 2 are very different. So the deep sea ecosystem at a fixed station can be changed. And finally, I can summarize the strengths and limitations of EDNA metabolic coding. Strength is it is very economical. Something certain amount of water is enough, but it needs consideration of sampling methods. And it is not invasive, no destruction of habitats, no harm to organisms. And it is not taxonomic expertise. It does not require no taxonomic expertise. Taxonomic assignment is automatic and needs no expertise if there is sufficient reference sequences. You can see that our reference database consists of about 60% cover deep sea. 60% of deep sea fishes around Japan, and there are many limitations. For now, we can perform EDNA metabolic coding for only fish and decapod crustaceans. And second, eDNA is simply an indirect genetic marker shed from macroorganisms, and there is no information on size, maturity, and in many cases, sexes, and so forth. And in case of deep sea fishes, it needs a proof of concept. What does eDNA detection of species means? It simply flows from other places or it means the detection means species species occur at that place. Thank you for hearing.